Hi, in this second video about protein structure, we'll continue our discussion about protein structure. We'll talk about 4 levels of protein structure that scientists talk about when discussing protein structure, that being primary, secondary, tertiary, and quaternary. We'll spend a fair bit of time talking about 2 special types of secondary structure, alpha helix and beta sheet. We'll give examples about how related protein sequences can produce related 3-dimensional structures, and in turn the proteins will have related functions, not necessarily the same. We're going to go into greater detail about binding interactions for protein, and then we're going to talk about a structural way that proteins control their activity. So we're already going to start talking about sort of a high level of regulation of the protein, and it's pretty amazing about the way it happens automatically. I think you'll like that. Okay, so the first level of protein structure is called the primary structure, and all it is the linear sequence of the amino acid. It's nothing besides just telling the amino acid sequence. So the linear primary structure of this tetrapeptide is methionine, aspartic acid, leucine, and tyrosine. Secondary structure of a protein is common regular foes that we'll see over and over. And 99% of what we're going to talk about are alpha helices and beta sheets. These are common folding patterns that proteins adopt. This is a couple different pictures of an alpha helix. As you can see over here on the right, it's a right-handed helix. And in a little more detail in section B, there are 3.6 amino acid residues per turn. Each amino acid has an angle of about 100 degrees, so 4 amino acids would be 400 degrees, so that's a little bit more than a full turn. And 5.4 angstroms between a turn, so that's the pitch of the helix. One of the really important things that happens if you look at the backbone atoms, the carbonyl oxygen and the hydrogen attached to one of the nitrogens, these form a series of hydrogen bonds. So as we talked in the previous lecture, an alpha helix is a straightforward way of satisfying all the hydrogen bonding possibilities of these backbone atoms, and that's one of the reasons that it has such a high propensity for appearing in a folded protein structure. Alpha helices are one simple way that proteins can cross lipid bilayers, membranes. And that's because you can have a string of about 20 amino acids here that are only the 10 hydrophobic amino acids, so that they can interact with the hydrophobic lipid membrane tails. If you could not bury a charged amino acid here in this portion of the alpha helix. So that means you can computationally identify regions of proteins that are likely to cross the membrane. Whenever you see 20 amino acids in a row that are hydrophobic, then you have a good hint that this is one possible folding structure it could take, and so you might guess that it's a membrane protein. Another thing to point out, that the amino acids above and below those 20 will often be hydrophilic, because they're interacting with the hydrophilic heads of the lipids and also the aqueous environment out here. Now, let's imagine what happens, and this sort of anchors the protein. Let's imagine what happens if you're moving the peptide either up or down here. As soon as you start to pull it down a little bit, you'll be pulling these hydrophilic residues down into the hydrophobic interior, and you're going to this hydrophilic amino acid is going to resist being pulled down here. It's energetically unfavorable. So these hydrophilic residues will prevent the peptide from slipping downward. Similarly, these hydrophilic residues will not want to be pulled up into this so it's going to keep it from slipping upwards. So by positioning hydrophilic residues on the edge of this hydrophobic run, you are anchoring the alpha helix in the membrane in exactly the location you would like it to. Here we see another type of secondary structure, a beta sheet, schematized by arrows. In this case, it's an anti-parallel beta sheet with the direction of one going up. Again, this would be the amino end. This would be the carboxy end. For this strand, it would be the amino end. This would be the carboxy end, and then an amino end and a carboxy end. The beta sheets can also be parallel, where all these arrows are going in the same direction. This is the backbone atoms, the alpha carbon, the nitrogen, and the carboxylic acid, and they adopt this sort of pleated sheet here, and that they will line up. And once again, we see when we look at the hydrogen bonding between the carbonyl oxygen and the hydrogens on the amino groups, that they are all satisfied. So this is, again, a common way of solving the problem of having all the backbone hydrogen bonds being satisfied. So you also notice that you have two sides of the sheet. You have some R groups that are below, also here, below the sheet. And then you have another set of R groups that are above the sheet. So you can, just like with alpha helix, you could have one side being hydrophobic or hydrophilic. In this case, the beta sheet can have different characters on the different sides. Moving up a level of organization is the tertiary structure, and this is the complex final fold of a monomer subunit, where that includes all the alpha helices, all the beta sheets, the way the various domains, sort of sections of proteins, fold next to each other, and also disordered regions that are just sort of random loops or linker regions. So the tertiary structure is the final folded structure of a monomer, and the last level of protein structure is the quaternary structure, and this is the interaction of different subunits. Here's a monomer green subunit and a monomer red subunit, and the way they interact gives rise to this overall protein complex that is called quaternary structure. In this case, you can see that the green subunit and the red subunit look very similar. If they are in fact similar, this could be a homodimer. These could also be related molecules, heterodimers, if there are some slight differences between the red subunit and the green subunit. Now there's one additional aspect of protein structure that we haven't discussed, and that is disulfide bridges. Cysteine residues have this thiol group right here. It's kind of like a hydroxyl, but sulfur, the atom below oxygen in the periodic table, forms a sulfhydryl group instead of a hydroxyl group, and two sulfhydryl groups can form a disulfide bond, and then you have a covalent bond interacting two parts of the chains. The other types of structural elements that we talked about, electrostatics, hydrophobic interactions, hydrogen bonds, those are all through space. So this is forming a new covalent bond. These cysteine bonds are reversible. And in fact, these work as good redox sensors of the environment if you have a reducing environment, you're going to have free sulfhydryls. If you expose the protein to air, they're going to get oxidized. They will form disulfide bonds. So this is a natural way in which proteins can sense the environment and change its structure as a result, and then perhaps change its function. We saw earlier in the course that DNA sequences could be lined and that indicated evolutionary relationships. The same thing can be done with protein sequences. There are two protein sequences here, the human sequence and the Drosophila, a fruit fly sequence. The first of 50 amino acids are listed. And in the yellow, there's this sort of a comparison band. If two amino acids are identical, like glycine and glycine, you will see the letter for the amino acid glycine being indicated. If there's a similarity, like for instance aspartic acid and glutamic acid, they're not the same, but they're very similar, so they'll get this plus sign here. If things are very different, like lysine and proline, there will be nothing in the space there. And this slide also goes on to say that there are certain sequences that are characteristic of a large number of proteins, and those were highlighted in green. And so you might expect there to be more agreement between these two proteins in these regions if they have the same function, but that's a minor point. Now, if proteins have different but similar but related protein sequences, they might have similar folds, and this is an example of that. These are two proteins, a class of proteins called serine proteases. The regions in green indicate that they have identical sequence. The regions in gray here indicate different sequences, but as you can see, the overall shape is pretty similar, and especially this red line here is the active site. That's the enzyme, the binding pocket where the enzyme will bind as substrate. This region is particularly similar for these two proteins, but as you can see in the loops here, there are some differences, and those are important but not critical. These slight differences in the protein shape will mean that these enzymes will process different proteins. Elastase will break down a specific protein called elastin, whereas chymotrypsin is a protein that will break down many different proteins. So we went from related sequence to related three-dimensional structure to related but slightly different function. So one last point about protein structure and to show how the cell will regulate protein structure as a way of controlling protein function. We saw earlier that proteins have both complementary shapes and charges here at interfaces, that the shape is in essence the way that the cell is correctly positioning these charges so that you get a strong interaction. And that can be a protein-protein interaction over here, or a protein-ligand binding in which the ligand is a small molecule. So this is highlights over here from crystal structures about the way proteins bind a small molecule called cyclic AMP. And what's impressive here is that there are 7 different interactions. Hydrogen bonds, 6 of them, 2, 3, 4, 5. There's actually 2 here, so 6. And an electrostatic salt bridge to ensure that very few other molecules would be able to utilize or take advantage of all 7 of those potential interactions to bind there. So this is a specific binding pocket for this molecule. And what's interesting is that the amino acids that contribute to this pocket come from many different places in the protein, and it's only by creating the folded protein with the proper shape that you bring these different amino acids into close proximity to create the binding site for cyclic AMP. Okay, so now utilizing those principles to control protein function, one of the standard ways cells do that is by a mechanism called feedback inhibition. And here the cell is metabolizing different molecules, A, B, C, and X, Y, and Z. And so when you have molecule B, you have 2 choices. You can go down and make molecule X, or you can make molecule C. So what we're going to see here is how the cell automatically shuts down the process to making molecule Z, and so that all of the intermediate molecules B will go on to become molecule C. Now this is an enzyme complex that's involved with the production of a molecule called CTP. And If the cell has lots of CTP, it doesn't want to make any more. And if there's a lot of CTP around, there's also some that's available for binding. And the process of CTP binding to the enzyme changes the conformation. Here you see it's more open and now it's closed up, is going to make this enzyme inactive, turning it off. And so you'll, because you have so much CTP, you will not be making more. And so then just to show these on the two slides together, CTP is like the molecule Z here, and this enzyme complex is the enzyme that converts B to X. But if you have a lot of CTP, you don't want to convert molecule B to molecule X anymore. And so, but since there's a lot of CTP, it's around, it's thermodynamically going to bind the protein because there's so much of it around, and that will have the effect of shutting down this enzyme. So all the amount of molecule B will get converted to molecule C. And that's a pretty impressive way of starting to make complex regulatory decisions for these proteins based entirely on thermodynamics in a very logical way. So we'll see other examples of how cells use allostery to regulate gene transcription, but this is a way of using allostery to control protein function. Okay, that's it for this one.